Ma'am holds an MBA in HR management and completed the EPG program in health economics in 2023. Ma'am has joined in the Kerala Medical Education Service in 2010 with over a decade of service at multiple GMCs across Kerala. Ma'am has completed in basic and advanced courses in medical education, currently a faculty member at the NMC Nodal Center for Faculty Development, Research Enabling, Environmental Subcommittee of the institution, IEC Bharat Hospital Kottayam, IRB Caritas Hospital Kottayam. Ma'am is a life member of various organizations like IPS, NAPT, ISCR and SASP, and has been actively engaged in academic and professional forums. Ma'am has been awarded the best paper in SRIPS conference in 2008 and 2009, and recently Ma'am has won the best paper in Apticon 2024 in AIMS Raipur. Ma'am has over 43 publications, various index journals with 370 citations with an H index of 12. Ma'am currently pursuing PhD under Kerala University of Health Sciences, and been an active resource person in various local and national conferences and webinars. Over to you, ma'am, for a wonderful session.
Thank you, Doctor, for that introduction. Can I share the slides?
Yes, ma'am. Ma'am, please click hide button. Yes, ma'am.
Yeah. So at the outset, I would like to thank the Department of Pharmacology at SRM for inviting me to give a talk in this session. And I also would like to take this opportunity to congratulate the whole team of SRM who have been continuously working for many years for conducting this ASPER 2025. So I am sure all of you are very tired listening from the morning. So is there anyone who is still energetic and tell which drug is implicated in this slide? So this is a patient who developed severe hypersensitivity reaction following initiation of an antiretroviral drug. I think all of you are drained off, and let me continue. So this drug is implicated in causing severe hypersensitivity reaction is abacavir. Now, why is it relevant? Why we have started with this case scenario? Ideally, the pharmacogenetic testing for HLA-B57, which is related to severe hypersensitivity reaction of abacavir, should have been done before initiation of the drug. So coming to our topic, I'm Dr. Danya, working at Government Medical College, Kottayam. And for the next few minutes, I'm sure you all want to leave. So we will quickly go through the presentation. We'll be dealing about the topic, Pharmacogenomics, Frontiers in Drug Therapeutics. So we have only a few headings. So at the end of the session, I want all of you to remember that for some drugs, we need to do pharmacogenetic profiling so that we can translate the concept of pharmacogenomics into clinical practice. Secondly, you should be able to write the essay which will be asked for the exam, and you should be able to cite at least 5 examples of pharmacogenomics that I'm sure all of you would be knowing. So this is the overview. What is pharmacogenomics? What are the genetic variability? We'll be talking about only a few. What is the application of pharmacogenomics in drug therapeutics? That will be combining and talking. The technologies driving pharmacogenomics, barriers and strategies in implementation, ethical and financial issues, and finally, conclusion. So we all know pharmacogenomics is exploring how a gene is interacting and causing variability in the response of drug, whether it is pharmacodynamically or pharmacokinetically. So we are moving away from the concept of one drug fits all to the concept of personalized medicine. And this is because each patient's genetic profile differs from one person to other. There will be optimal responders, especially in whom the genes would be normal variants. And if it is a mutant variant, either heterozygous or homozygous, it could result in suboptimal responders, non-responders, or even hyper-responders, which would present as adverse drug reaction in the clinics. So this Food and Drug Administration is specifically labeling the prescription drug with pharmacogenomics. So this is a table which is available in the FDA site, and you can see that in this table of pharmacogenomics, the drug is specifically labeled based on whether it will cause a drug exposure and clinical response variability, the risk for adverse events, genotype specific dosing, mechanism of drug action, polymorphic drug target and disposition genes, trial designs. So these are the different indications or different labels in using which the FDA is labeling the pharmacogenomic relation to a drug. Now, as of September 2024, 395 drugs which are meant for human prescription has this pharmacogenomic labeling. Now, another big database is the pharmacogenomics knowledge database. Now, pharmacogenomics knowledge database, you can see that the FDA annotations as of yesterday is 481. The drug label annotations is 1,188. That is, it is getting from sources other than the FDA. And there are 209 clinical guideline annotations and specifically 263 curated pathways. So when we go to the site, you can see that this is the pharmacogenomics pathway sites. And these are the different evidence-based clinical curated pathways in which pharmacogenomic pathways are depicted for pharmacokinetic as well as pharmacodynamics of a drug with relevant potential pharmacogenomic associations. So this is the pharmacogenomic knowledge database where you can find details about all the annotations till now present in this world that is related to pharmacogenomics, with even if it is FDA approved or non-FDA approved, and also specific clinical curated pathways. So coming back to our presentation, the introduction of pharmacogenomics, we all know, will start with a definition. And that definition is how pharmacogenomics, that is genetic makeup of a person, is interacting with the drug. So the drug gene interaction will translate into pharmacodynamic effects. It could be an increased efficacy of the drug, a decreased efficacy of a drug, or increase in the presentation of adverse drug reaction. Or it could be polymorphism, that is pharmacokinetic polymorphism, and that will translate into pharmacokinetic variations. So because this is a PG revision class, if someone could unmute and tell, it would be good, or else I can tell. So this is a 29-year-old patient who has been diagnosed with epilepsy. And because you are in an ideal setting, which is providing drugs free of cost, and we only have old generation anti-epileptics like carbamazepine and phenytoin in your institution. So this is the patient's genotyping that has already been done. So based on this table, can you comment on the drug which you will initiate for this patient? Can anyone unmute and tell, or shall I proceed? So I think there is no response from the other side, so I'll be proceeding. So from this, we can see that there are two pharmacogenomic associations that are evident here. One is the HLA association. And this HLA association we have already mentioned at the initiation of our slide, that is abacavir hypersensitivity, which is related to HLA-B57. Now, does anyone of you know whether it is the HLA-B or the HLA-A that is more specifically attached to the severe cutaneous adverse drug reactions of carbamazepine? It is HLA-B15 allele that is associated with severe cutaneous adverse drug reactions of carbamazepine. HLA-A star 31 allele is associated with DRESS. Okay, and there is less incidence of DRESS if the patient is having HLA-B star 40 allele. Now, that is translation of this pharmacogenomic association into pharmacodynamics. What about CYP2C9? We all know that phenytoin is metabolized by CYP2C9, and the wild variant is the star 1 allele. Star 2 and star 3, if it is present as homozygous or heterozygous allele, they will be either intermediate metabolizers or poor metabolizers. So in such cases, there will be increased risk of development of toxicity. So if at all you are going to start this patient on a drug, that would be phenytoin, and the dose of phenytoin would be reduced. So this is one classical example that you can remember from the central nervous system, that is CYP2C9 star 2 and star 3, which is causing alteration in the phenytoin metabolism, causing high levels of phenytoin and thus causing phenytoin toxicity. Association of HLA-B is also seen with phenytoin, that is HLA-B star 15 allele is also causing severe cutaneous adverse drug reaction in both phenytoin and carbamazepine. Lamotrigine is also causing severe cutaneous adverse drug reaction associated with HLA. Now, the other examples that I would like to cite from the central nervous system is the dopamine receptor, that is the D2. We all know antipsychotics act through the D2 receptor, and a mutation in the DRD2 gene that will decrease the efficacy of antipsychotics. Now, CYP2D6 is another drug metabolizing enzyme, and CYP2D6, if it is having a mutation which is causing a non-functional allele, then what will happen to the drug levels? There will be an increase in the drug level, and even for newer antipsychotics like risperidone or aripiprazole, the patient will present with extrapyramidal symptoms and tardive dyskinesia. Now, another example is the long allele, that is a repeating polymorphism of the long allele of the serotonin receptor. We all know that the SSRIs modulate the serotonin, and action on the serotonin receptor, it is increased, and the incidence of adverse effects is decreased if the patient is specifically having the long allele of repeating polymorphism instead of the short allele. Now, another important point I want to tell regarding the drugs used in the central nervous system is codeine. Now, codeine, you all know, is it an active drug or a prodrug or an active drug which is converted to an active drug? Codeine is an active drug which is converted to another active drug, and that is morphine. So, if the patient is having CYP2D6 polymorphism in which the patient is an ultra rapid metabolizer, then what will happen? The patient will go in for respiratory depression. So these are the few examples that I want to cite from the central nervous system. One is the anti-epileptics, second one is the SSRIs, the antipsychotics, and codeine. Now coming to the next case, that is case 2. Now, the same patient, because he is a poor metabolizer, we decide to start him on a low dose of phenytoin. So the dosing of phenytoin is like this. If the patient is having a star 3 variant, then we need to reduce the dose by half of the dose should be given. 1/3 of the dose should be given. And if the variant is star 2, we need to give half of the dose. Now we are going to talk about the use of anticoagulants. So as I already mentioned, even though genotype testing is done, this is an ideal setting, a government hospital where only warfarin is available. So would you give warfarin in this patient if there is a suspected thromboembolism? You have already started the patient on low levels of phenytoin because it is a star-2, star-3 variant of CYP2C9. Will you start on warfarin? So we know that the warfarin metabolism is complex. It is determined by so many clinical factors, dietary factors. There is drug interaction between so many drugs, and again the condition of the patients, the concomitant diseases. Now, how pharmacogenomics plays a role in warfarin? Warfarin dosing. So this VKORC, that is vitamin K epoxide reductase enzyme, that is coded by this VKORC1 gene, and that is again here a mutant variant. The GG variant is the mutant variant, and the patient will have a decreased response to warfarin. That is, if it is a GG variant, there will be warfarin resistance. Now, the other genes that you need to look for while starting a patient on warfarin would include the CYP4F2 genes, the GGT genes, and also genes which are encoding for protein C and protein S. That is, protein C and protein S deficiency that could result in thromboembolic events if the patient is started on warfarin. So there is an algorithm based on warfarin therapy. So when I joined service in 2010, I wanted to do warfarin profiling, and at that time I had written a protocol, but it didn't see light because as I already told, in a resource poor setting, till now the conversion of research work into an actual outcome is a distant dream because of the lack of infrastructure, because of lack of trained staff, and so many other things. So I had to drop that study because by the time everything was in the institution, there was a treatment algorithm already published based on this thing. So even in 2010, there were so many studies, but now there are actual prediction models based on which warfarin dosing is done. So as we are Indians, we will directly go into this. What is done usually is dosing based on clinical factors, especially in our institution. We don't go for a genomic profiling. But if the data of VKORC1 and CYP2C9 star2 and star3 genotype is available, then we have to use the pharmacogenomic algorithm. So let us go into this site. So there is a site for pharmacogenomic warfarin dosing. And once you enter the details, you can enter various details like the age, gender, ethnicity, and also the concomitant medications, the personal habits, and also the availability of genes. As I already told, the GG variant is warfarin insensitive. Then CYP4F2, the CC is the normal wild type, and the GGC that is the wild type is CC, and CYP star2, star3, and they are the mutants. Homozygous mutant is TT. And again, CC is the homozygous mutant in the case of CYP2C9 star3. So thus, if we completely fill this, then we can estimate the warfarin dose. So this is actually a translation of the pharmacogenomic profiling into actual clinical practice. Whether it is done here or not is a distant dream, but it is not very distant now. Now, another example I want to cite from the cardiovascular system is regarding which drug? That is clopidogrel. So the P2Y12 inhibitors, clopidogrel, ticagrelor, all these, the main enzyme that is metabolizing is CYP2C19. Now, CYP2C19 is relevant in the context of clopidogrel specifically. Can anyone tell why? Because clopidogrel is a prodrug and it needs to get converted to its active drug. So if a person is having a mutant CYP2C19, That is, if it is a star 2 or star 3 allele. In the context of phenytoin, we told that if it is a mutant variant, it would be poor metabolizer. So what will happen if the patient is a poor metabolizer? Unlike in the case of phenytoin, where there was phenytoin toxicity, here there is decreased efficacy of the drug. Why? Because clopidogrel is a pro-drug. And there are several clinical trials, especially the tailored antiplatelet therapy after PCI, that is the Taylor PCI trial. There has been evidence generated, especially during using the Bayesian analysis. They have found that if we do pharmacogenomic profiling before starting clopidogrel of CYP2C19 mutants, then it will help in decreasing the major cardiac adverse events. Now, another mutant is CYP star 1 star 17. So, CYP star 1 star 17 is a hyper responder. But in the case of clopidogrel, what will hyper response do? It will produce an increase in the effect of the drug. So, that is about clopidogrel. Now, Dr. Nidarshan was going on telling about the different transporters, which transporter mutation is associated with the statins, that I'm sure all of you would have, you would be knowing. That is because Dr. Nidarshan was keeping on reinforcing all the transporters several times. So that is about the cardiovascular system. So from the cardiovascular system, please remember the examples of warfarin, clopidogrel, and statin, which can cause myopathy because of mutation in which gene? So coming to the next one, that is case 3. Case 3 and 4 are related to cancers. So a patient with non-small cell lung cancer is sent for genomic profiling by the oncologist. And before showing the profile of the result, that is the pharmacogenomic profiling, whether there was a mutation present or not, let us look into the different mutations that can occur in non-small cell lung cancer. So in a patient who is having non-small cell lung cancer, there could be a mutation in the epidermal growth factor receptor. And there can also be a mutation which is causing a fusion protein of the anaplastic lymphokinase as well as the EML4 gene. So when this occurs, if the patient is positive for anaplastic lymphokinase EML4 gene mutation, then the patient won't be usually having EGFR mutation. That is, they are mutually exclusive. So if the patient is positive for ALK EML4 gene mutation, there is no role in giving EGFR inhibitors, that is the tyrosine kinase inhibitors like gefitinib, erlotinib, afatinib, and osimertinib. And the ALK inhibitors include crizotinib, ceritinib, and alcitinib. So if the patient is having a mutation of the ALK EML gene, we have to give ALK inhibitors. And if the patient is having a mutation only of the EGFR gene, then we can go for tyrosine kinase inhibitors like gefitinib. So next is about breast cancer. So another epidermal growth factor is Herceptin. HER2 new is extensively expressed that in the breast cancer, and the treatment of breast cancer, for that we need the profiling of this receptor, that is HER2 protein needs to be profiled. And if the patient is positive for HER2, we would go for treatment which drug? Which all monoclonal antibodies are given if the patient is positive for HER2? We'll be giving trastuzumab, pertuzumab, or azotrastuzumab emtansine. So this is one classical example you can cite where pharmacogenomic has been implemented into decision making whether to give a drug or not. So the epidermal growth factor expression, there is lots of HER2 expression, and the treatment response is also somewhat related to the HER2 expression. So if the patient is having a mutation in the BRCA1 or BRCA2 genes, what will happen? If the mutation is in BRCA1 or BRCA2 gene, the enzyme that is repairing the DNA will be not proper, and the tumor will resort to repair of the DNA, single stranded repairs. So the PARP, that is adenosine ribose polymerase inhibitors, olaparib, niraparib, and rucaparib. So they inhibit the utilization of the alternate single-stranded repair pathways, which will result in lot of unwanted DNA molecules, and that will initiate the apoptosis. So if the patient is positive for HER2, we will go for monoclonal antibodies, trastuzumab, pertuzumab, azotrastuzumab. And if the patient is positive for BRCA1 and BRCA2, we will go for polyadenosine ribosyl polymerase inhibitors like olaparib, niraparib, and rucaparib. Another example that already Dr. Nidarshan has mentioned in his presentation, that is regarding the SN38 mutation of which drug, and also our DPD mutation of capecitabine and 5-fluorouracil. So coming to the last clinical scenario that also Dr. Nidarshan has mentioned. So before going to that, I was telling about the testing for the receptor, mutant receptors. That is, we can see that in 1998, the FDA approved trastuzumab for treatment of breast cancer. And in the same year, FDA has approved the utilization of trastuzumab testing for breast cancer. So this is an area where the discovery of mutation or the treatment or discovery of mutation followed by the targeting of therapy and its diagnosis have got simultaneous permission from the FDA. Again, in 2012, the drug pertuzumab was marketed. So, and the FDA has approved the trastuzumab, pertuzumab diagnostic kit as well. And again, in 2013, the azotrastuzumab emtansine was approved for the treatment of breast cancer, along with its testing. Lapatinib also, its use and its testing has been approved. So before the patient gets treatment, it is mandatory to do this test. Before we initiate a patient on antiretroviral therapy, it is mandatory to do HLA-B57 before starting abacavir. But it is not mandatory to test CYP2C19. It is good if we test CYP2C19 polymorphism before starting clopidogrel. Now, this also Doctor has already mentioned in the previous presentation regarding cystic fibrosis. So, he told that if there is a mutation in the delta 5F08, what will happen? That will produce an abnormal protein, a misprotein that will be engulfed by the endoplasmic reticulum, and that will not be present in the cell membrane. So, what should we give? We should give what type of drug should we give? We should give correctors or potentiators. We should be giving correctors like lumacaftor. And if there is a mutation in the cystic fibrosis TR gene at G551D, then what will happen? There is a problem with the opening and closing of the gating channel. So what will this drug do? Ivacaftor is a potentiator and it will help in gating of the channels. So this is another area where pharmacogenomic profiling has been translated into actual clinical practice. And as the doctor already told, we have several combinations of corrector as well as potentiator. The one combination that has been approved is the lumacaftor-ivacaftor combination. So these are a few examples I want all of you to remember. First one, what was it regarding the central nervous system? Anti-epileptics, that is carbamazepine, phenytoin, antipsychotics, D2, CYP2D6, SSRIs, as well as codeine. Now coming to the cardiovascular system, warfarin, clopidogrel, and which was the SLC mutation that was affecting, that was causing myopathy, that I presume all of you know. And in the oncology, I want you to remember about the non-small cell lung cancer, where there is EGFR mutation, ALA mutation, and also the BCR-ABL, that is BCR-ABL expression, that fusion protein is seen in which cancer? That is seen in chronic myeloid leukemia and also in gastrointestinal stromal tumor. And which drug are we talking about? We are talking about the use of imatinib, a tyrosine kinase inhibitor. So that is about chronic myeloid leukemia. And for the treatment of breast cancer, the Herceptin estimation, as well as the use of diagnostic testing, and also the breast cancer BRCA1 and BRCA2 mutations, and the mutations of cystic fibrosis. So I'm sure these few examples all of you would remember. So are drug modulations the only therapeutics in the context of pharmacogenomics? No, pharmacogenomics is not really explored by the drugs, the drug gene interaction. There is a high level increase in the gene editing through zinc finger nucleases, the TALENs, as well as the CRISPR. And there is also development of the medical softwares, especially based on our genetic profiling or the genomic profiling softwares, which will target behavioral changes and help in diagnosis as well as mitigation of diseases. So as I told, one dream in my career is to do a pharmacogenomic testing. So in 2019, I had given for another testing, but still it has not seen light. So who are the stakeholders who are going to implement pharmacogenomic testing? So the regulators who authorize the test, the hospital leadership, the payers, that is the insurance companies, play a crucial role in implementation of the pharmacogenomic profiling. Of course, the developers, which include the Drugs and Therapeutics Committee, the laboratory, and also the IT cell. And finally, the users, that is we, the doctors who are going to prescribe the drugs, and also the patients for whom the drug is being prescribed. So these are the stakeholders in implementing that is actual testing to clinical practice. Now, we can do two types of pharmacogenomic profiling. One is the preemptive genotyping and the reactive point of care genomic profiling. So from the figure, it is evident even before there is a diagnosis, we profile everything, and then we go for a pharmacogenetic consultation, or when we need the report, we look back into the pharmacogenomic profiling that is already there in the database, and based on that, clinical recommendations. That is called as preemptive genotyping. And the other one is the usual one, where first there is a clinical evaluation followed by pharmacogenomic profiling. That is called as the reactive point of care testing. Now, what is the actual status? Even in the developed countries, there is a lot of issue, because preemptive genotyping, the insurance companies or the payers don't support much. And it is the reactive point of care testing that is usually happening, except in where there are specific guidelines that the testing should be done before the initiation of the drug. From what we heard a few minutes back, we know that there are only a very few examples where it is a must to do a pharmacogenomic profiling, even though there are lots of prescription drugs for the humans. Now, what are the challenges in implementation of pharmacogenomics into clinical practice? As I already mentioned, the major thing is lack of funds. That is the expense of genetic testing and its implementation. That is whether we should channelize the money that is available to the public, or that is available with the government for pharmacogenomic testing, when there are so many other pressing needs. That is the cost benefit analysis. We have to think about that when implementing the pharmacogenomic testing in our institution. And of course, there is a huge knowledge gap among the healthcare professionals, especially whether there is any clinical utility for pharmacogenomic profiling, and actually whether it will interfere with the clinical workflow. Imagine a patient is coming for initiation of therapy, and he needs to wait for 1 week or 1 month before actually starting the therapy. So that interference with workflow is another major problem that is causing barrier to implementation of pharmacogenomics. Then the complexity of pharmacogenomics, the limited research in the diverse population, and the lack of standard testing, all these are barriers. Now, what are the solutions? So there is only one and only solution, that is increase education. That is, we have to incorporate pharmacogenomics, even though pharmacogenomics is there in the undergraduate curriculum, in the postgraduate curriculum, and when we are actually practicing, the actual concept of pharmacogenomics is not being imbibed. And for that, we require inclusion of pharmacogenomics in curriculum as well as research. Then second thing is insurance, availability of funds. Why the patient has to spend so much money from his pocket, that is the out of pocket expense, if the insurance company is not ready to pay, especially for preemptive diagnosis, it would be a huge issue. And other strategies including diverse populations in the pharmacogenomics research, implementation of standardized guidelines, and increased awareness and promoting genetically guided prescription. So regarding the implementation guidelines, in India, we don't have any regulatory guidelines specifically for pharmacogenomics. What we are using is the clinical pharmacogenomics implementation guidelines, CPIC guidelines, and also the safety guidelines by the same. So what are the ethical considerations while performing pharmacogenomics or pharmacogenomic related research? Like any other research or any other test, the patient consent and data privacy is of prime importance. But other things include potential discrimination based on the genetic background, especially when we are collecting large scale pharmacogenomic data for a research work, or for profiling, for preemptive testing. Then who is going to own this huge database? That is the public versus corporate ownership of the results. Now, the randomized control trials, which are trying to find out a solution for rare genomic indications, what about its post-trial access? Will it be available to everyone equally? After it is marketed, what about the accessibility of treatment based on cost? These are the pressing ethical issues in pharmacogenomics. So coming to the last few slides. So the time of a single nucleotide polymorphism estimation is gone. Now what we do is next generation sequencing and integrated bioinformatics tool, which will help in finding out the large details that we get while doing pharmacogenomic testing. And the emerging trend is, of course, the integration of artificial intelligence and machine learning, which will help not only in finding out the drug target, gene mutation, also for the development of drugs. So in conclusion, I want to tell that pharmacogenomics is not a distant myth, but a hope for the medical future, so that we can develop better drugs. We can shun the concept of one size fits all, and we can also tailor the dose of the drug. And also advanced screening, lifestyle for lifestyle and ecological changes, improvement in drug discovery and approval, and reduction of healthcare. So thank you. So this was the SLC 01B1 star 15 allele that is implicated in adverse effect related to which drug? Which groups of drug? This transporter. So thank you.
Thank you, ma'am. It was a wonderful session. When gene speaks, transporters respond, and pharmacogenomics listens. It's where biology meets tailored therapy. It was a wonderful and enthralling session, ma'am. And dear delegates, we are coming to the end of day 1. If you have any doubts, please share in the WhatsApp group that will be shared to ma'am.
Thank you.
Thank you so much, dear delegates. It's an end for day 1 session. Tomorrow we will meet on the same at the same time. The link will be shared in the WhatsApp group. Thank you all. Thank you so much, ma'am.
Thank you.